This protocol enables continuous measurement of genetic circuits in E. coli and the calculation of the signal variability and signal-to-noise ratio in individual bacterial cell to assess circuit performance.
The main advantages of this technique are that it is easily adaptable to different strains and labs, requires minimal training and no special equipment, and is relatively inexpensive. Noise has a significant role in determining the functionality of biological systems. Currently, there is an interest in building large-scale gene circuits. These systems can be inhibited by noise.
Be sure to dry the samples well to keep the shell pads at 4 degrees Celsius for as long as possible and to cut and apply the pads patiently and gently.
To prepare the bacteria for the analysis, inoculate a single colony from a transfected E. coli culture, for example, a circuit with a GFP reporter, in a glass tube containing 5 milliliters of LB broth supplemented with the relevant antibiotics. Grow the cells at 37 degrees Celsius and 250 revolutions per minute in a shaking incubator for 2 hours until the broth is cloudy. At the end of the incubation, spin down 1 milliliter of dilution solution in a 2 milliliter mini microcentrifuge and transfer 30 microliters of bacteria and the entire volume of dilution solution into a new 2 milliliter tube. Then incubate the culture for 1 hour with shaking, before seeding 40 to 60 microliters of the suspension onto M9 culture plates. To prepare the bacteria for the